Hello, everyone. So this is a continuation of chapter 2 lecture. We just finished talking about non-membranous organelles. So let's go ahead and get into the organelles that have a membrane around them. So so we have this guy here that's called mitochondria. So this is zoomed in on mitochondria here. So this is something that produces ATP. This is the powerhouse of the cell. So what does that mean? That means that it's going to be an organelle that produces ATP. So when we say powerhouse of the cells, what we really mean is that it's making that ATP. ATP is a chemical that contains the energy. So it's the powerhouse of the cell. It's going to take your food, it's going to break it down, convert it to energy, and the molecular bonds from the food stick it into this molecule called ATP. So ATP is the currency of all life. You take ATP, you can do a lot with it. So this guy here is going to be the same size as bacteria. So mitochondria size is the same as bacteria. And this is going to have a membrane. It's going to have 2 layers of membranes, and inside here it has even some DNA in the nucleus. So in the mitochondria. So it's going to have mitochondrial DNA that's in there. So that's called let's say it's called mDNA, mitochondrial DNA. So it's going to have mDNA. So this has mDNA. So this is a separate DNA that you find in the nucleus. This is in the mitochondria. So this is one of the reasons that we think the mitochondria came from bacteria, because they're same size as bacteria, same shape as bacteria, and there's also DNA in it. So why would it need DNA? It's probably because the organism was living outside of the cell way back then, and then somehow it got inside the cell and it just worked as a symbiosis, which means they work together. Maybe it's giving it energy and it's giving food in return. So, symbiosis is a relationship that's both two species benefiting from each other. So, nucleus itself has the chromosomes in it, and we will talk about that in a second. So, nucleus contains chromosomes and it's a control center for the cell. So here's a close-up of what mitochondria looks like. And it's really small. It's an organelle about the size of the bacteria. It has a bunch of extra folds. It has this membrane, inner membrane, outer membrane. It has a bunch of these extra folds on the inside that give it extra surface area for enzymes in order to make ATP. So this helps us to generate lots of ATP, versus if your bacteria and the bacteria converting glucose into ATP wouldn't be very efficient. On the other hand, if you were to use the mitochondria, you can generate anywhere from 34 to 36 ATP compared to the bacteria, which can only make 2 ATP per glucose. That's pretty efficient. All right, so let's head into the nucleus, and this big guy here is called the nucleus, and so that's the nucleus, and inside this nucleus is this something called nucleolus. So all organisms have a nucleus. Organisms that don't have a nucleus, they're called prokaryotes, and we are called eukaryotes because we have a nucleus. Eu means true, so we have a true nucleus. We're considered to carry nucleus, and karyo means nucleus, eu means true. So we're eukaryotes. On the other hand, bacteria and archaea, they're prokaryotes, means these are the organisms that evolved before the nucleus. So what's in the nucleus? The nucleus is going to house all your genetic information. So all your DNA, all of your nucleotides, those A's and T's and G's and C's, those are the nucleotides: adenine, guanine, cytosine, thymine. So they're going to go and link up and turn those into something called chromatin. So here you have the nucleus, and there is this chromatin in the nucleus. And when you keep zooming in, it's going to look like this, chromatin, and then when you unwind it, it's the double helix DNA, and these color-coded things are the nucleotides, the A's, T's, and G's and C's. So I don't know if you remember, but we got 23 pairs of chromosomes, so 46 chromosomes, and that's where 23andMe, the genetic testing, came from, because in a healthy person, we get 23 pairs of chromosome. One pair comes from mom and, one comes from mom and one comes from dad, so making a pair. And that's what's basically going to be inside the nucleus. You have this little dark ball, and that's called the nucleolus. And connected to the nucleolus, we have all these folds. So here, that's the nucleus. You have the nuclear envelope around it, and you have these folds that are connected to it. And these are called endoplasmic reticulum. So we have the endoplasmic reticulum network of hollow tubes. And we learned about the in general bio. So this endoplasmic reticulum, you can call it, maybe you've seen it called ER. So on the quiz or the exam, do not call it ER. You have to write the whole thing down. And there are two kinds. So you have one that's connected directly. That's called rough endoplasmic reticulum. And then you go a little bit further out, then you have smooth endoplasmic reticulum. The reason this is called rough endoplasmic reticulum is because there are ribosomes on it. They are speckled all on the surface here, and you got these ribosomes, and then under the microscope, it looks kind of bumpy, and that's why it's called rough ER. So what's the function of rough ER? We know that ribosomes produce proteins. So what's happening here is that you have inflammation from the nucleus, and that's causing enzymes to produce chemicals, which will go through the ER, and it'll go through the endoplasmic reticulum and leaves the endoplasmic reticulum and goes to the next phase, which is going to a bunch of stacks that are called the Golgi apparatus. This is the Golgi apparatus. So you have your smooth endoplasmic reticulum on this side, and you have your rough here, and there are no ribosomes on this, and there are ribosomes on this part of the ER. So this is rough and this is smooth, and then you have more folds that look like kind of stacks, and this is the Golgi complex or Golgi apparatus. So it's going to take the proteins that were initially produced by the ER, then absorbs it through, and then it modifies them and packages them into vesicles and kind of sends them out through exocytosis, and then they're taken up through endocytosis, so all the different processes. So these chemicals will leave, and some of them, they come out and turn into vesicle that'll merge into the membrane. And if you remember, that's, those chemicals go into the interstitial fluid of your bloodstream, and then these could be hormones, could be enzymes, and then it will go to the other parts of your body. So Golgi produces these vesicles, and they house all these different kinds of enzymes. So you have Golgi apparatus that's modifying the proteins. Okay, so then there are these other ones, lysosomes, that contain cellular digestive enzymes. So they can either, turn into peroxisomes that contain catalysts to break down hydrogen peroxide. So there are two kinds, lysosomes. They are going to be digestive enzymes. They're like garbage disposal. So lysosomes and peroxisomes, they're garbage disposals over yourself, and that's because you have some waste products. The waste products are going to get gobbled up by these, because they have these enzymes that can do that. And then it gets thrown out into the interstitial fluid and the bloodstream. And again, lyse, the word lyse means to break. So lyse means to break. So it's going to break down the waste products and dispose them. So these are the ones that break up enzymes or molecules. They have the enzymes to break down the molecules. And peroxisome, on the other hand, are going to have a chemical in there that's called hydrogen peroxide. And this is also to break stuff down. So there are two types that you got lysosomes and peroxisomes. They both behave kind of like a garbage disposal. They're going to gobble stuff off that your cells don't need anymore. And that's going to be inside the cell. And then it's going to basically get thrown out. So let's put this all together. So here, this is showing the ribosomes, giving it that rough appearance. You have the nucleus, and then you have connected to it, you have these folds, which is the rough endoplasmic reticulum. You also have the smooth endoplasmic reticulum. They look like kind of stacks here, stacks. So putting it all together, here we have something called the endomembrane system. So endo means inside. So endo means inside. And so you have your nucleus here. So there's the nucleus. And then in this system, there's information in the nucleus that's going to generate molecules in the ER. So that's the endoplasmic reticulum. So the information is going to go into the, I'm trying to make an arrow, ER. ER will then send those molecules to the next thing that's going to be Golgi. So it's sending the information to Golgi here, and or Golgi apparatus that's going to modify the molecules, and these molecules will immediately leave the cell. So they're leaving the cell here. So they're going to leave the cell, and those molecules turn into, something called lysosomes. These are the ones that contain the digestive enzymes. So if any waste products needs to be excreted out, or peroxisomes. This whole process here, the whole system here is called the endomembrane system, where endo stands for inside, and this is a membrane. So it's on the inside of the membrane. So if I were to ask you about endomembrane system, you should write something about nucleus, ER, Golgi apparatus, and vesicle. So basically, in order which stage, in order where in which things leave the cell. So like that, in that direction. So now we're going to go ahead and go to the next slide. So last thing we're going to cover are called examples of cell junctions. So basically how cells stick together. So you have one cell here, and then you have another cell here. So we have one cell, another cell, another cell. They're sticking together, but they're sticking together in different ways. So what do they use to stick together, basically? So they stick together using these things called junctions. And if you have, if they have a hole in it where you stick to a membrane together, there are holes over here where molecules, so there's a hole there, some space, but then they're sticking together here. So if there is a hole, basically, where they're sticking together like that, those are called gap junctions. And these are the holes or the gaps. So here you can see them here as well. And then on the other hand, you got tight junctions. And this is something that's like where you pretend that you have two pieces of paper and you staple it together. That'll be a tight junction. That's not the only kind. So within this category of tight junction, you have these things called desmosomes. You have spot desmosome and you have hemidesmosome. So the difference is that if it's a really good one, it's a spot desmosome. If if it has only one protein, then it's a hemidesmosome. So there are two different kinds. So let's look at these two. So let's look at the gap junction first. So you have these gaps, but the cells are still sticking together. So these are called gap junctions. And these are something that can be found in your heart, that are found in your heart. So what's happening here is there is an electrical signal that's traveling through these gap junctions. It'll go from one cell to the next, to the next, to the next. And you're able to do this because you have gotten the gap junction that has holes in it for the signal to go through. So, and then there's going to be one that's called spot desmosome. So this is a spot desmosome, and you'll see it has a protein on both sides. So this is called a spot desmosome because you have protein on both sides, but then we also talked about hemidesmosome. This is zoomed in on hemidesmosome. There's only one protein on one side and has these roots in it. So this is hemidesmosome, and this is spot desmosome. And then there is, let me see if there's a tight junction. Yeah. So there is a tight junction here. It looks like two papers were stapled together. So that's a tight junction. So these are two membranes that are stapled together, making the tight junction. So that's how cells stick together. So you can go ahead and stick these cells to each other using these different types of junctions to make tissues. So these junctions stick the cells together, and that makes the tissues. And that's chapter 3. So chapter 3 is how to form tissues. That's going to be about tissues. So now we have finished the introduction to anatomy, and this chapter was review on the cells. Next one, we're going to take what we learned, and we're going to start building the tissues. And then after that is when we're going to go into the organ systems. So that's it for this chapter. Thank you for listening.